This is our presentation on Phyllorhiza punctata.
So the Phyllorhiza punctata is the white-spotted jellyfish. It's in the kingdom Animalia, the phylum Cnidaria, the class Scyphozoa, and its genus is obviously Phyllorhiza, and the species is P. punctata.
So the white-spotted jellyfish is a rhizostome scyphozoan. It has a diameter of 45 to 50 centimeters, and it undergoes polyp and large medusa stages. The rhizostome medusa have a large hemispherical bell, which is an umbrella-like structure, and that's marked with crystalline inclusions, which gives the white-spotted appearance.
So the body plan of the white-spotted jellyfish has 8 radial canals that lead to the stomach, along with thick 8 transparent oral arms. So these right here are the oral arms. They lack marginal tentacles, but they have minute tentacles around the lips of the mouth. In the adult P. punctata, oral arms are 2/3 of the bell's diameter, and there's tons of filaments containing stinging cells that are found on the upper surface of the arms. There are 8 rhopalia, which are sense organs that have the eye and the statocyst, which is used for maintaining balance on the margin of the bell, and they have 14 lappets, which are rounded lobes that allow the bell to flex. The polyps of the P. punctata are around 2 millimeters in diameter, and the stems are 3 to 5 centimeters long, and they have conical bodies.
So the P. punctata lives in warm coastal waters. It favors lagoons and salt-tolerant waters, and they also prefer to live in the shallow regions.
So for nutrition of the white-spotted jellyfish, they often have zooxanthellae, which are symbiotic algae, so they're able to provide the jellyfish with nutrition via photosynthesis. They provide carbon in exchange for nitrogen. These zooxanthellae are what gives the bell of the jellyfish the brown-colored appearance, and this mode of nutrition is often sufficient enough for the jellyfish to survive. Low salinity conditions, however, are not ideal, as the zooxanthellae are absent in these conditions, and the jellyfish must obtain nutrition from zooplankton instead.
So for their reproduction, they undergo normal cnidarian reproduction, which alternates between asexual reproduction during the polyp stage and sexual reproduction during the medusoid stage. In the case of the P. punctata, a scyphozoan, reproduction is often dominated by the medusoid stage. So for this species, the sperm is shot from the males into the water and are collected by the females via their mouthlets, and then filtered to the reproductive organs. The resulting planktonic planula larvae settle on the bottom to grow, and this is known as the scyphistoma stage. From there, the larvae undergo the process of budding and form additional scyphistoma or develop into a strobilia, which produces a benthic form and asexually reproduces to make epiphyrae, which are the young medusae. So for the general distribution, they are found in northern Atlantic waters, and they have expanded through the Gulf Stream in warm waters. They are originally found in Port Jackson, Australia, but have since migrated.
So the effects of the invasiveness of the white-spotted jellyfish include ecological and economic impacts. So some of the ecological impacts are impacting the other organisms in the area. So the medusae blooms may consume large extreme amounts of zooplankton, which takes away the available nutrients for larval fish and invertebrates. Populations in Laguna Joyuda, Puerto Rico, have decreased the abundance of zooplankton significantly enough to reduce fish recruitment. And some of the economic impacts are that their blooms are so massive that fishing nets can be clogged, and because of the extreme consumption of zooplankton and eggs, that may also affect fisheries. P. punctata also might have the ability to affect tourism. It's not really a deadly sting that comes from them, but it creates an unpleasant area for people to swim and a lot of dead jellyfish in the area, which just isn't ideal for tourists. But impacts on fisheries seem to be more significant than the impacts to tourism.
So the primary research article that we used for this presentation, it was Seasonal Patterns in Size and Abundance of the Phyllorhiza Punctata, an invasive Siphomedusa in coastal Georgia. The study was originally submitted by Dr. Peter Verity, who unfortunately passed away before publishing, so the editor of Marine Biology published his work in honor of him. And with this paper, the researchers' goal was to determine if expansion of the white-spotted jellyfish had occurred in coastal Georgia waters from 2007 to 2008.
So over the 2 years of the study, 4 pairs of sounds and adjacent shelf sites were visited in 4 to 6 week intervals. So this figure on the right demonstrates the sampling stations along the Georgia coast. So the inner shelf stations are indicated by the circles and the adjacent sounds are indicated by squares. There were no previous reports of the jellyfish in the coastal or shelf waters prior to 2007. So they decided to study this area to see if they had migrated.
And with figure 1b, this is an image of the Gulf Stream path which they thought that the jellyfish traveled. So as you can see, it starts down over here and then loops its way around the base of Florida and up towards Georgia and the Carolinas.
So for the sample process, researchers utilized a small boat that was run at a slow speed, about 3 kilometers per hour, for 15-minute periods and sampled in visual transects on calm days. All of the P. punctata that were observed were numbered, and this visual transect allowed the researchers to see a 5-meter wide strip next to the boat that was only 0.5 meters deep. So the result was an 800-meter long survey transect and a 5-meter wide swath at 0.5 of a meter depth. So this image is just kind of an idea of what a visual transect is. This is the way they were able to observe the jellyfish. Each station measured the bell diameters of 10 P. punctata to the nearest centimeter so that growth could be compared among each sample date. And species identification was based on the morphology and the previous photos of the P. punctata from the Gulf of Mexico and Atlantic coast. However, the taxonomy and population relationships remain uncertain.
All right, so for their results, they sampled within 2 years, and they only found 444 medusa, and that showed that the densities were greater at shelf stations, and the differences in medusae sizes found at the shelf stations were insignificant. Many of the P. punctata medusae samples were collected from like May 2007 in the northernmost shelf stations, and samples in 2008 were collected from all shelf stations, but greater concentrations were found at the innermost stations.
So when they measured the temporal abundance patterns from the sound stations and the inner shelf surface waters, they found that more medusae were seen from June to November at inner shelf stations, but they were rarely spotted in sounds. Again, on this figure, you can see the squares represent the inner shelf stations and the circles represent the sounds. The medusae density increased during the spring and summer, but decreased in the fall and winter throughout the 2 years.
So this table demonstrates the temperature, salinity, medusae density, and the diameter at the inner shelf stations that were recorded between May 2007 and December 2008. So the densities of the medusae were significantly greater at shelf stations than at sound stations in both years. However, differences between shelf and sound stations in medusa sizes were not significant. Environmental factors did not effectively describe this difference in the densities, as salinity was only slightly lower in sound stations compared to shelf stations, and temperature showed no significant difference in results. And because so few medusae were seen at sound stations, the scientists decided to only focus on shelf station analyses moving forward.
So in this table, we can see the temperature, salinity, the medusae density, and diameter at sample stations from May to November. And we see from this chart that there were higher total densities in 2008 than 2007, and more medusae were counted in 2008 due to the number of stations being too small to count in certain months within 2007. Overall, the medusae size was larger in 2007 than 2008, with the exception of November, and significantly more medusae were counted from July to November 2008 than in 2007. Water temperatures were also slightly warmer by roughly 0.5 degrees Celsius, and salinity levels were higher by roughly 1.5 to 2.5 in 2008. This may be a contributor to size differences throughout the 2 years, but P. punctata were found to be absent and rare in the winter months, so due to this factor, data was not collected in all months due to the winter weather.
So this figure describes the swimming bell diameters of the jellyfish. So it was found that the mean bell size increased in diameter until November 2007, and it began to decline in October 2008. The smallest medusa collected was in May 2007 with a diameter of 10 centimeters, and they found that the maximum mean diameters were 30 centimeters for both years. The growth calculations from max changes in diameter. Growth was also calculated from the maximum changes in bell diameter, so they were from 10.3 to 29.3 centimeters between May 20th and October 6, 2007, which is roughly 140 days, and from 16.4 to 32 centimeters between the 25th of May and 29th of September 2008, which is roughly 128 days. And within that, they found all lacked symbiotic zooanthali.
So figure 4 shows the inner shelf near surface water temperatures, and we can see from this graph that warmer temp and higher salinity has been correlated with the increased number of medusae and asexual reproduction of medusae. Springtime conditions may explain the difference between the 2 years in the number of P. punctata medusae in Georgia. NOAA temperatures demonstrated that in 2008, February to March was warmer than in 2007, and temperature and salinity measurements demonstrated that in July through November of 2008, it was slightly warmer and slightly more salinity in July through October than both rates in 2007.
So for discussion, the data from this study does not necessarily demonstrate P. punctata is established in all Georgia waters, but it suggests that they're only established in particular waters. And the critical step was to establish annual populations in the northern Gulf of Mexico, and they realized that the jellyfish could have been introduced as early as 1993, but only began blooming in the 2000s. They also discovered that non-random transport mechanisms were implied based on the sequential chronology of the P. punctata's expansion, which suggests that they spread via oceanographic and not ship-mediated transportation. And they followed the loop current, which flows clockwise around Mexico and went through the Gulf Stream, which supports the theory that they transported from the Atlantic locations and went to the Gulf populations.
So they also found that the data doesn't necessarily demonstrate, again, like she said, that P. punctata is established in all Georgia waters. Evidence supports that the medusae were transported from the established populations in Florida within travel time of 8 to 17 days, based on the distance between Jacksonville and Savannah, with an average wind speed of 10 to 20 centimeters per second. And this matches up with life cycle and growth rates of the P. punctata. And it's not totally clear why they didn't show up in Georgia sound shelves. Highly turbid waters might have hindered visual sampling, and the polyp populations were not present. Data about the life cycle was not collected, which is central to understanding the success of all jellies and their ecology. Seasonal patterns of the Medusae P. punctata are similar to those found in Brazil, Puerto Rico, and Australia. And when comparing temperature and salinity changes that occurred in Australia to those in the study, the environmental conditions similarly affected the Medusae. We correlate the Medusae numbers and an increase in asexual reproduction, and the temperature data recorded in the spring supports the idea of Medusae being produced locally. Higher salinity and temperatures reflect greater influences from the Gulf Stream, and differences arose in growth rates when comparing data from populations in Georgia and Puerto Rico. But the Medusae in Puerto Rico were larger than those in Georgia, which may reflect the food availability, temperature, and presence of zooxanthellae.
So for future studies, they are going to look at the behavior, susceptibility to predation, the life cycle, reproduction capacity, and the physiology of the jellyfish, because they want to know more of a basis as to what's going on with them. And then it will aid in establishing a basis for further ecological success.